Good morning and good afternoon, everyone. So I appreciate the opportunity to share some of our work with you, targeting the interaction between RNA-binding protein HuR and FOXQ1 suppresses breast cancer invasion and metastasis. So first, what is HuR? So the full name of HuR is human antigen R. It is a member of the HuR or embryonic lethal abnormal version-like protein family. So it is also known as HuA and ELAVL1. HuR preferentially binds to the mRNAs bearing U or AU-rich elements in their 3 or 5' untranslated region. Once binding to these mRNAs, HuR mainly stabilize and promote the translation of them. HuR predominantly localized in the nucleus, but its function of stabilizing and promoting translation is linked to the translocation from the nucleus to the cytoplasm. It is found that elevated cytoplasmic HuR correlates with advanced stage, high grade, and poor outcomes in several types of cancer, including breast, prostate, colon, and pancreatic cancer. So here is an example in breast cancer. So the authors determined the cytoplasmic HuR expression in 133 breast cancer patients, and they found that the patients with high cytoplasmic HuR has reduced distant disease-free survival compared to those has low or non-cytoplasmic HuR. So we also did a retrospective expression of the HuR in breast cancer patients. We found that the patients with higher grade has more positive standard cytoplasmic HuR. We also try to compare the patients with metastasis and without metastasis. So you will see that the patient with metastasis has slightly higher positive standard cytoplasmic HuR, but because this the number of the metastasis patients is few, so the p-value did not reach the significance. We also find that the patients with high HuR has lower overall survival and also the distant disease-free survival, which is consistent with the previous slide show. To study the HuR function in breast cancers, so we use the CRISPR-Cas9 technology to knock out the HuR in a panel of breast cancer cell lines. So here I use the MDAMB231 cell line as an example. So here we have 2 clones with HuR knockout. So these 2 clones, they grow much slower compared to the parental cell and the control cells. And they cannot form colony. And also they have fewer invaded cells in the invasion assay. So we also inoculated this clone 1 to the mice and found that these mice cannot form tumor at all. So then how does HuR promote tumor genesis? Here is a list of the HuR target genes. You will find that these target genes are implicated in almost all the cancer hallmarks: the proliferation, survival, evasion of immune recognition, angiogenesis, invasion, and metastasis. So HuR may serve as a diagnostic, prognostic, and therapeutic target. There are 2 strategies can inhibit the HuR function. So one is that it can bind to the HuR and then move HuR back to the cytoplasm, back to the nucleus. So here the MS444 is a small molecule reported that can have this function to inhibit the translocation of HuR. So another strategy is use the small molecule that direct binding the HuR, which will disrupt the binding of HuR with its target mRNA, so which will leading to the decay and the reduced translocation of the mRNA. So that's what the strategy we used. And to identify the HuR inhibitors, we generate a fluorescent polarization assay for high throughput screening using the HuR protein and the RNA oligos of several reported HuR targets. So here we use BCL2, MSH1, XIP. These three are representative HuR targets. Totally, we screened 23,000 compounds and with a hit rate about 0.8%. So the one of the top hits, the KH3, today I'm focused on, is from a screening of 2,000 compounds. Here is the structure of KH3. And at the same time, we identified compounds which have very similar structure with KH3, but it is negative in the screening. So later we use it as a negative control for the studies. Okay, so first we used several biochemical assays to validate that KH3 is a HuR inhibitor. So on the top two panels are the FP and alpha assays, which shows that KH3 can disrupt the binding of the HuR protein on all the arm1 to the RNA recognition motif with the MSH1 RNA oligo. So with a Ki about 0.8 or 0.7 micromolar, but it's a negative control. KH3B has a minor effect. On the bottom left is the SPR, which tried to confirm that the KH3 is directly binding to the HuR protein, has a dose response effect. On the right is the computational docking of the KH3 and the KH3B to the RNA binding pocket of the HuR. So you will see here, this part of the KH3, it can form two extra hydrogen bonds with the HuR protein, which explains why KH3 is significantly potent than the KH3B. Okay, so then we try to confirm that the KH3 can bind to endogenous HuR and also block disrupt the binding. So here we first use called a cellular thermal shift assay, CITSA. So we in the cell we heat the cells in different temperature and detect the HuR expression. So you will find that with the KH3 treatment, HuR is more stable. Okay, which confirmed that the KH3 can bind to endogenous HuR. We also use the 2IP or 2 pull-down assay, one called the RNA pull-down, one called the RNP-IP. So for the RNA pull-down, we use the RNA oligo to pull down HuR protein. So for the RNP-IP, we use the HuR protein to pull down the bound mRNAs. So here we show that the MSH1 RNA oligo can pull down HuR protein, but the amount of the HuR can be decreased by the treatment with the KH3, but not its negative control compounds. And at the same time, we show that these three mRNAs can be pulled down by the HuR protein, and then treated with the KH3, you will see a partially disruption of the binding. Okay, then we move to the cells to test the cellular activity of the KH3. So here is the cytotoxicity of the KH3 against a panel of breast cancer cell lines. You will see that the KH3 has potent cytotoxicity against them. So we also compare the cells, the two CD1 cells I mentioned before with HuR knockout. You will see these two clones with HuR knockout, they become less sensitive to KH3, which suggests that the cellular activity of KH3 is at least partially dependent on the HuR. So here are two movies. So on the left is treated with DMSO, on the right is treated with KH3. So you will find that when cells treated with KH3, the proliferation is inhibited. So here the cell proliferate very fast, but on the right, cells just grow very slow. And then you will see that cells start to die. So see here. There. Here the cells are pretty happy and healthy. Okay, so I mentioned before the function of HuR is stabilize its target mRNAs and also promote the translation. So, and then I test if the KH3 treatment can block these functions by testing the half-life of the target mRNAs and also the protein expression level. So here you will see that the half-life of all three mRNAs are decreased by the KH3, and also the protein level are also decreased. You will notice that the HuR expression level were not affected by KH3, as KH3 function as block HuR function instead of decrease HuR function. Okay, so we also did the migration and the invasion assay and to detect HuR KH3 function on the migration and invasion. You will see here treated with KH3 cell has less migration and also fewer invaded cells. So then we ask how KH3 inhibit the migration and the invasion. So we first did a qPCR array using the 88 human invasion and metastasis genes. And at the same time, we also did use the HuR knockout clones. We found that the KH3 treatment is very similar. The profiling is pretty similar to the HuR knockout clone, and we found that the CDH1, which the encoded protein called E-cadherin is an absolute marker, is increased. So we also use the CDH1 promoter reporter assay and confirm that KH3 can increase the expression of CDH1. And then we ask, how does KH3 promote CDH1? Does this relate to the HuR function? So then we did two RNA-seq. So one is called RIP-seq, so which can identify the HuR direct targets. Another is the RNA-seq treated with the KH3 or the control. So we identified a gene called FOXQ1. It is a direct HuR target. At the same time, it is one of the top genes decreased by KH3. So several papers has reported that FOXQ1 can promote the EMT and also the metastasis in breast cancer by negatively regulate of the CDH1. So we first determined the expression of FOXQ1 and E-cadherin in the human mammary epithelial cell and also a panel of the breast cancer cell line. So you will see that these two proteins are negatively correlated. And then we use these two IPS I just mentioned to confirm that KH3 can disrupt the binding of the HuR and FOXQ1. So here we use the FOXQ1 ARE site to pull down HuR protein, and then the treated with the KH3 that this pull down can be decreased. So this at the RNPIP here, so compare with the IgG, we'll see a huge enrichment so which confirms that FOXQ1 as the HuR targets, and then their interaction can be decreased by the KH3 treatment, but not the negative control. So since HuR binds to the 3' UTR of FOXQ1, so we use the FOXQ1 3' UTR reporter assay and confirm that KH3 can inhibit the FOXQ1 3' UTR activity. And then we ask if the FOXQ1 contributes to the HuR function in the cell invasion. So we did overexpression of FOXQ1 in the knockout cells. So you will see that the cells with overexpression of FOXQ1, the invaded cell numbers is increased compared with the control. At the same time, we detect the CDH1 expression level. So in the knockout, you will see the CDH1 expression is increased, but without FOXQ1 overexpression, its inhibition, this induction is attenuated. So which suggests that the FOXQ1 at least partially contributed to the HuR function in promoting cell invasion through inhibition of the CDH1. So similarly, we did FOXQ1 overexpression and same time treated with the KH3. So you will see that here the KH3 can decrease the invaded cell numbers, but with FOXQ1 overexpression, this inhibition is almost completely rescued. So at the same time, you will see the CDH1 expression can be decreased by the FOXQ1 expression, which also confirmed that the inhibition of the FOXQ1 is the mechanism how KH3 inhibits cell invasion. So then we moved to the in vivo study. Here is the 231 orthotopic model. So we found that with the KH3 single treatment, they can inhibit this tumor growth about 60%. So bottom is the protein expression in the tumors treated with the KH3 and control. You will see a decrease of FOXQ1 and also an increase of the E-cadherin, which is the protein of the CDH1, which is consistent with our previous finding in the in vitro. So we also did experimental metastasis model. So using this 2-LMP cell, the 2-LMP is an isogenic cell line we generated from the MDA-MB231 formed lung metastasis in the mice. So it is compared to the parental 231, it is more aggressive. So we use this cell line as our model. So we use bioluminescent imaging to monitor the tumor initiation and the progression in the lung. So here is representative images. You will see the progression of the tumor in the lung. So here on the top left is the initiation of the lung metastasis. You will see treated with KH3 can delay the initiation of the lung metastasis. And also on the right, you will see the overall survival also can be improved by the KH3 treatment. So to summarize what we are finding here, so we identified KH3 as a HuR inhibitor. It can disrupt the binding of HuR with FOXQ1. So FOXQ1 can negatively regulate CDH1, then promote EMT and metastasis. So there is also other report about the FOXQ1. It can promote LAMA4, which can promote the ECM degradation. So here we also have some data show that the KH3 treatment can also decrease the LAMA4. So another mechanism for the FOXQ1 is to promote the ECM degradation to promote metastasis. So we also found that the KH3 can disrupt the binding of HuR with beta-catenin, which can inhibit the metastasis suppressor CD82 then promote EMT. I didn't talk much about this today. So definitely there are other targets of HuR can promote the metastasis, but we have not identified. Okay, so I would like to thank our lab members, the current members and the previous members, the people at the KU core facility, and also our collaborators at the KU and other universities, and of course the grant support. So, okay, I thank you for your attention. I will stop here and be happy to take any questions.